So, uh, today I am going to discuss with you about the structure and types of your RNA, or you can say them as ribonucleic acid, because it consists of a ribose sugar. Now, in these notes, I have taken from internet sources, different internet sources. So let us study now. So, structurally speaking, ribonucleic acid or RNA is quite similar to DNA. Whereas DNA molecules are typically long and double stranded, RNA molecules are much shorter and typically they are single stranded. Why they are shorter? Because during the processing of RNA, those sections of RNA which are not required for the translation process, they are eliminated out through a process called RNA splicing. Okay, you will in detail you will able to learn from this chapter of RNA splicing in molecular biology classes, that RNA composed of different segments of introns and exons. Okay, the exons are the essential part, whereas the introns are non-essential part required for the translation process for protein production. So these introns are removed out through a proper process known as splicing, and that's why the RNA molecules are quite shorter compared to DNA. And basically you know that RNA is single strand, not double strand, so it does not actually follow the Chargaff's rule, which states that the amount of adenine present in the DNA is equal to the amount of thymine, and amount of guanine present in the DNA is equal to the amount of cytosine. It simply means that total amount of purines in the DNA is equal to the total amount of pyrimidines. So it does not follow this Chargaff's rule, this RNA molecule. RNA molecule perform variety of roles in cell, but they are mainly involved in the process of protein synthesis. So this RNA mainly involved in the process of protein synthesis and its regulation. And you know that during translation this RNA will be used as a template for the synthesis of a particular protein. Okay, now let us look at the structure of RNA. Let us overview. I have already discussed with you little bit about the what contains in the RNA. Same like DNA, but the pentose sugar that is found in the RNA nucleotide is actually ribose sugar. So RNA mainly is a single stranded and made up of ribonucleotides. These ribonucleotides are linked with one another by the same process by phosphodiester bonds. A ribonucleotide in the RNA chain contains the ribose sugar, which is a 5-carbon sugar, the pentose sugar, and it also consists of one of the 4 nitrogenous base: adenine, uracil, guanine, and cytosine. Yes, our RNA ribonucleotide does not contain thymine. Instead of thymine, there is uracil. A little bit of structural difference occur in the 5th position of thymine, that leads to the formation of uracil and a phosphate group. So generally, the ribonucleotide which makes your RNA same like DNA, but instead of thymine, RNA ribonucleotide consists of uracil. Little bit modification of the thymine at the 5th position of thymine leads to the formation of uracil, and it consists of a ribose sugar attached with glycosidic bond. This nitrogenous base attached with the pentose sugar with glycosidic bond, and the phosphate group also being attached at the C5 position, C5 prime position of your pentose sugar. So this makes a ribonucleotide, and one ribonucleotide attached with the another ribonucleotide by the same process by phosphodiester bond. So the subtle structural difference between the sugars give DNA added stability, making DNA more suitable for storage of gene. Okay, storage of gene, or you can say genetic information. So if you compare with the stability with the RNA structure, DNA is quite more stable than RNA because of its many functional groups which are modified. Okay, second, mostly RNA consists of a pentose sugar, and this that pentose sugar, that ribose sugar, in the 2' region there is a hydroxyl group which can interact with the bases uh, to form unusual structure which may hamper their stability. But in case of DNA, they have a missing hydroxyl group at the 2' position. That's why we name them as 2' deoxyribose sugar. Okay, and that does not involve in interaction with the bases in the alkaline solution. Okay, now, okay, so as it is being said that it makes the DNA more suitable for storage of genetic information, where the relative instability of RNA makes it more suitable for its short-term function. So RNA is basically involved not in stability but for immediate functions. We want to make immediately protein during that short period of time. So for that reason, from the DNA, transcription occurs. DNA uses the template to form RNA by the process of transcription by the help of some enzymes, specifically the RNA polymerase. You will see later on. So it is a short-term function. This RNA is based on short-term function. It is not based on stability, that it will remain for a long time in a cell. It will be quickly get degraded because they are less stable. Whereas DNA is more suitable for storing the genetic information. RNA specific pyrimidine called uracil forms a complementary base pair with adenine instead of thymine in the DNA. So RNA have a specific nucleotide, it has uracil in place of thymine, and this uracil can form hydrogen bond with the adenine nucleotide. Okay, now even though your let us see the structure of uracil that has been given here. Okay, so in the right hand side you can see the structure of thymine and the structure of uracil. Look at the fifth position. At the fifth position there is a group called methyl group, CH3 group in thymine, whereas in uracil there is no methyl group present. So this little bit modification makes it formation of uracil, which is found in the RNA, and adenine base pair with the uracil by 2 hydrogen bonds, okay, in your RNA structure. In the A figure you can see that RNA consists of a ribose sugar, whereas DNA consists of deoxyribose sugar. It's only that 2' position where you can see that the hydroxyl group is missing in deoxyribose sugar, whereas in ribose sugar that hydroxyl group is present. And that is the main reason that why RNA is not stable at alkaline pH, because this OH group at the 2' position can interact with the bases in alkaline solution, and that can hamper their stability. Okay, this is the structure of comparing between the RNA and DNA. RNA normally is a single strand, but even though it is a single strand, it sometimes folded. Many of the times it fold within each other. The you can say the intra coiling. Okay, within they are coiling itself. They are forming hydrogen bonds. Okay, so that's why it is written here. That even though in the last line, okay, even though RNA is a single stranded, most of the types of RNA molecules show extensive intramolecular base pairing. So RNA fold onto itself. It is a single strand, okay, it is single strand, but it fold on itself. Intramolecular, intra means within, they are folding, they are base pairing with each other between the complementary base pairing, okay, within the RNA strand, and it creates a particular or predictable three-dimensional structure of your RNA. As we have seen in tRNA, you will see in tRNA they are single strand, but they have fold on a particular positions by complementary base pairing. Okay, so here you can see the hydrogen bonding occurs in the B figure, that single strand RNA have folded onto itself by complementary base pairing, which is found within the single strand of RNA and involve and makes a three-dimensional structure. Okay, coming to the next part. Now the function of RNA in protein synthesis. It has been said that the cells access the information stored in DNA by creating RNA to direct the synthesis of protein through the process called translation. So translation is a process where the RNA is used as a template for the process of synthesis of proteins. And proteins within the cell have many functions, it builds up your cellular structure, it serves as an enzyme catalyst for cellular chemical reactions, okay, and on the based on that there are many types of RNA also which carries out the process of translation as well. Okay, so not only one type of RNA is involved in translation process, but there are different types of RNA, okay, which are involved in translation process for formation of protein. And those three main types of RNA are your messenger RNA, also written as mRNA, M will be in small letter, ribosomal RNA or rRNA, and transfer RNA or tRNA. So these three types of RNA are actually involved in protein synthesis, successfully in prepare the particular protein from that mRNA using them as template. Okay, so it is saying that the mRNA carries the message from the DNA, right? The information which is stored in the DNA, that information carries by the mRNA, because during transcription from the DNA the mRNA is prepared, and that mRNA carries that information and it controls all the cellular activities in a cell. Now suppose if a cell needs certain protein to be synthesized, then the gene are the particular segments of DNA which are the functional segments of DNA. So the gene at that time, suppose a cell require a particular protein, that we have to make this protein now immediately. So the gene, what happened, that segment of the gene responsible for the formation of that particular protein is turned on. Turned on means proteins help that gene so that they can undergo transcription in that site on in that segment of gene, and thus mRNA is synthesized through the process of transcription. Then the mRNA comes out from the nucleus, comes out from the nuclear membrane, okay, by the help of transport proteins found within the nucleus, and then mRNA will interact with the ribosomes found in the cytoplasm or in the surface of rough endoplasmic reticulum. They interact with the ribosomes and other cellular machinery to direct the synthesis of protein, okay, that it encodes during the process of translation. So simple thing, that the suppose the cell wants to make a protein, the gene responsible for that protein, they started transcribing with the help of enzymes like RNA polymerase, certain transcription factors are there, okay, and prepare mRNA from it. Now the mRNA is processed Okay, the unessential segments are removed away by the process of splicing, by the process of post transcriptional modification. You will see later on that the different modifications of RNA occur before being processed into the translation process, and then the mRNA comes out from the nucleus through the help of transport proteins. Then this mRNA will interact with the other cellular machinery, with the ribosomes as well, to direct the synthesis of protein that it encodes during the process of translation. So normally it's a very step-by-step method, and a little bit complicated also. Okay, during the process of translation, during the process of processing. Okay, so this is a normal diagram. You will see this diagram later on when I will discuss about the translation process in detail in later videos. You can see the two subunits here, the small ribosomal subunit and the large ribosomal subunit. They actually get assembled here during translation, but otherwise they are not assembled previously. You can see here that the amino acids are being formed with the help of the tRNA and mRNA. tRNA is another type of RNA that carries a particular amino acid on its terminal end, and this tRNA have sequences which are complementary with the sequences found in the mRNA, known as codon and anticodon. You will see later on what are codons. Actually, codons are normally 3 nucleotide long element, and each codon made up of 3 nucleotides that will code for a particular amino acid. These codons are complementary with the anticodons found within the tRNA, and they interact with these codons, and they carry the amino acids into the large ribosomal subunit site, where they interact with each other to form a peptide bond between the amino acids. And in this way, protein formation, the primary structure of protein, which are the linear chain of amino acid joined by peptide bond, is formed. Okay, many interactions occur, like small ribosomal subunit first interact with the mRNA with the help of its RNA, known as 16S rRNA or 23S rRNA in case of large ribosomal subunit. So you can see that with these RNA are mainly involved, the types of RNA they are mainly involved in translation process. Okay, so these rRNA and tRNA, if you compare these three things between rRNA, tRNA and mRNA, out of them rRNA and tRNA are quite stable. They are quite stable types of RNA, and in prokaryotes and eukaryotes, these tRNA and rRNA encoded in the DNA, then copied into long RNA molecules, and they are cut to release small fragments containing individual mature RNA species. So it is saying that in prokaryotes and eukaryotes, these tRNA and rRNA are encoded in the DNA, then they are copied into long RNA molecules, cut, and then they are cut to release small fragments containing individual mature RNA species. Now in eukaryote synthesis, cutting and assembly of rRNA into ribosomes takes place in the nucleolus region of the nucleus, but these activities occur in the cytoplasm of prokaryotes. Neither of these types of RNA carries instruction to direct the synthesis of polypeptide, but they play important role in protein synthesis. So simply means that if you see the rRNA structure, the rRNA are the components of ribosomes. Ribosome is made up of two subunits, the large ribosomal subunit and the small ribosomal subunit. And and within this ribosomal subunit, we contain important components of rRNA. So rRNA is a component of ribosomes. Okay, and the tRNA, they are specific types of RNA which carry suitable amino acids. Both of them are stable, but these rRNA and tRNA, none of them carries the instructions to direct the synthesis of polypeptide, but they play an important role in your protein synthesis. Okay. Now ribosomes, as we know, they are made up of RNA. What type of RNA? rRNA and proteins. And rRNA means ribosomal RNA, and this, as the name suggests, rRNA is the major constituent of ribosomes, and it composes about 60% of the ribosomes are made up of this rRNA only, and they provide the location where the mRNA binds. So rRNA actually just like a map, okay, just like a direction or GPS, just like that. It directs the ribosome that where it has to bind to initiate the translation process normally. Even after initiation, during peptide bond formation also, it somehow directs. And the rRNA ensures proper alignment of mRNA, tRNA and ribosomes, right? So rRNA also interacts with the tRNA also during peptide bond formation within the ribosomes. It has also a enzymatic activity. There are some RNA in the ribosomes which have enzymatic activity, that is peptidyl transferase activity, and they catalyze the formation of peptide bonds between two aligned amino acids during the protein synthesis. Okay, so these rRNA of the ribosome, they also have a enzymatic activity, which is the peptidyl transferase activity. They transfer the peptide bond, or they catalyze the formation of peptide bond, okay, and then transfer also between two aligned amino acids carried by the tRNA during protein synthesis. Okay, so this was about rRNA. Specifically rRNA structure has not been given. Okay, we just see them in the form of small segments found in the small ribosomal subunit and large ribosomal subunit. Now in case of tRNA, tRNA is the third main type of RNA, which is one of the smallest RNA that we found, made up of only 70 to 90 ribonucleotides long, and carries a specific amino acid to the site of protein synthesis on its extreme end, and it is a base pairing between the tRNA and mRNA which allows the correct amino acid to be incorporated into the polypeptide chain being synthesized. tRNA consists of an anticodon sequence. I have said already what is codon sequence. Codons are 3 nucleotide long unit which codes for a particular protein. Now when this codon sequence are recognized by anticodon sequence, they match with the codon sequence, and what the codon sequence code for that particular amino acid that is carried by the tRNA, okay, within the ribosome. Now if there is somehow mutations in the tRNA structure, mutations means variation in the nitrogenous bases due to some chemical agents, due to some physical agents like ultraviolet radiations, x-rays or gamma radiation. Some chemical agents are there like your ethidium bromide, like some carcinogenic agents you can say, okay, that cause them to mutate. That means modification of the bases occur in the tRNA structure. So if any mutations in the tRNA or RNA occurs, it will hamper the translation process. It will cause the translation process to occur, but there may be possible that the anticodon sequence may get altered, and that codes for wrong amino acids and wrong polypeptides are formed, creating a problem during protein synthesis. So all the three things are required during protein synthesis. We require mRNA that carries the information to code for a particular protein. We require tRNA also, because tRNA carries that particular protein, particular amino acid sequence, and it matches with the proper codon found in the mRNA by complementary base pairing. And they it is also called oval base pairing, because it will be different of type of base pairing occurs in the mRNA, between the mRNA and tRNA, you will see later on. And it carries that amino acid, where rRNA is also required, because it actually it is a component of the ribosome, and it allows the ribosome to specifically bind with where the ribosome has to bind with the mRNA for initiating the translation process, as well as it also catalyze the peptide bond formation. It has a peptidyl transferase activity, as I have said, that rRNA of the ribosome also have a enzymatic activity, peptidyl transferase activity, and it catalyzes the formation of peptide bonds between the two aligned amino acids carried by the tRNA during protein synthesis. So all three of them are required, without which translation process is not possible. Okay, so you can see the structure of tRNA now. On the left figure, it is a linear model of tRNA. Okay, and the big circle that you can see, it is actually carrying an amino acid, and the name of the amino acid that it carries here is glutamic acid. Okay, it has been shown in a beautiful way that how it carries the glutamic acid. It is binding to the extremities of the tRNA, that is at the 3' end. It carries the glutamic acid. Normally it can carry any amino acids depending on what type of anticodon sequence are found within the tRNA. We can see at the here CUC, it is the anticodon sequence for the tRNA which is carrying glutamic acid. Now generally amino acids can attach with this tRNA with the help of a process, with the help of certain chemical reactions called amino acylation of tRNA. It is a two-step process where amino acylation of tRNA occurs for attachment of particular amino acid to the tRNA at the 3' end. Okay, so just look at the structure of tRNA. It is a single-stranded, right? It is a single-stranded structure, but it has folded on particular locations. Okay, and this intramolecular base pairing occurs within the tRNA that gives the tRNA its secondary structure. Okay, and you can see a complementary base pairing is occurring, not complementary base pairing, you can say oval base pairing, or show how it is base pairing with the nucleotides, ribonucleotides in the mRNA. Okay, during protein synthesis. Now generally we have shown you three nucleotides, and 3 nucleotides makes a codon actually that codes for a particular amino acid. Like for example GAG in the mRNA codes for glutamic acid. So the tRNA that is carrying glutamic acid, it is complementary base pairing with the GAG codon of mRNA within the ribosome, and in this way it carries the glutamic acid. In this way perfectly that what the codon encodes, that particular amino acid comes and binds with the help of mRNA. Now on the right figure you can see this is also the structure of tRNA, but on a 3-dimensional view. Okay, you can see the acceptor arm, that carries the amino acid. You can see the anticodon loop also here. Okay, so now a little bit difference between the mRNA, rRNA and tRNA has been given. mRNA structure, I have not discussed the mRNA structure here in detail, but I will try to discuss later on, or within this video I will try to discuss. So mRNA is short, unstable, single-stranded RNA that correspond to gene encoded within DNA. So mRNA are very short structure, unstable. They are not quite stable for a long time. They will be easily degraded after translation process, and single-stranded RNA that correspond to a gene encoded within DNA. Okay, what is the function of mRNA? It serves as a intermediary between DNA and protein. So mRNA actually serves as an intermediary between the DNA and protein, and it used by ribosomes to direct the synthesis of protein it encodes. So it is acting as a template for the synthesis of protein, because the ultimate aim of our central dogma is to create protein from that particular DNA that has that information. So RNA is acting as a mediator. It is transcribed from the DNA and translated to form the protein. RNA is quite longer than mRNA, and they are very much stable RNA molecules. They are the part of ribosomes, and about 60% of the ribosome constituted by these rRNA. And rRNA is also involved in your protein synthesis, because rRNA ensures proper alignment of mRNA, tRNA and ribosome during protein synthesis. It catalyzes peptide bond formation. It has a peptidyl transferase activity. It can catalyze peptide bond formation between two amino acids during your translation process. So from proper binding of ribosome to the mRNA up to your peptide bond formation, peptidyl transferase activity, all are done by the rRNA. Okay, so they are also playing an important role during translation. Obviously tRNA also play an important role. tRNA are quite short, about 70 to 90 ribonucleotides in length. They are also stable RNA molecules with extensive intramolecular base pairing. That is, it's a single strand, but they have folded on certain regions by complementary base pairing within the found within the single stranded RNA. And tRNA carries amino acid at its 3' end, okay, which is a T, and it's called a tRNA binding site, and it allows that proper binding with the mRNA by codon anticodon base pairing. Again I repeat, codons are the 3 nucleotide elements that codes for a particular amino acid. Each mRNA have series of codons from start codon to stop codon. Okay, and the zone between the start codon and stop codon together called as open reading frame. So open reading frame, or ORF, consists of series of codons right next to each other. They do not overlap, and each codon codes for a particular amino acid, right? tRNA recognize these codons with their own 3 nucleotide element called anticodon. So tRNA have anticodon, that complementary base pair with the codon sequence in the mRNA that codes for a particular amino acid, and it carries that particular amino acid by complementary base pairing, or you can say oval base pairing with the mRNA. In this way tRNA also plays an important role. So it carries a correct amino acid to the site of protein synthesis in the ribosome. Now one more thing I have not shown, I have not discussed with you. You can see that tRNA structure. Now this model of tRNA structure that you can see on the left hand side, it is actually the clover leaf model. Clover, CLOVER, clover leaf model of tRNA. Now it has many loops. You can see from the left up to the right there are almost about 4 loops are present, and we name them and name these loops. The loop which is found on the extreme left is actually called D loop. D capital D loop, because of presence of dihydrouridine. The loop at the bottom where you can see the anticodon sequence are complementary base pairing with the codon sequence in the mRNA. This loop is called anticodon loop. Then a small loop that we can see just after the anticodon loop towards the right side, that is a variable loop. Now this loop name as variable loop because it varies between the tRNA. Presence of this loop ensures stability of the tRNA. Then you can see right next to the variable loop at the right side you can see a loop, and that loop is known as T psi U loop. Okay, T psi U loop. Now psi symbol, the Greek letter, the Latin letter symbol psi represents just like a you know the psi symbol, psi. The psi symbol represents here the pseudouridine. You will see later on the video on the next lectures that psi represents pseudouridine, and pseudouridine is an unusual base. Just like dihydrouridine I have said, pseudouridine is also an unusual base. Then just after this T psi U loop there is no loop present, but at the extreme where the amino acid has been attached, that site is known as the amino acid attachment site, also known as acceptor arm. A acceptor arm, right? And acceptor arm consists of a unique sequence called ACC. Okay, here it is written GCC, it will be ACC. Okay, that site is responsible for attachment of amino acid in a process called, in a two-step reaction known as amino acylation of tRNA. So you can see here the structure of tRNA, then you will see the structure of mRNA in detail on the videos. Okay, so later on I will try to discuss with you about the structure of your mRNA. From here I will discuss with you, I will try to discuss. I hope you have understand this video lecture. Okay, I'll provide you the notes just after the completion of this nucleic acid chapter. Okay.